So before we get into the actual contractions, let's take a couple seconds here and review the actual histology of the muscle cell and the components therein. So when we remember the histology of the muscle cell, we have to remember is that it's going to be the peripherally polynucleated tissue with the cylindrical arrangements of myofibrils with the cytoskeletal filaments proteins contained within it. There are distinct structures within the skeletal muscle cell itself that's going to be heavily involved with the coordination of the contraction. This includes the transverse tubules and the cisternae of the sarcoplasmic reticulum, known as the triad. In the transverse tubule, what we're allowing to do is we're allowing for the depolarization moment to pass from the membrane itself into the central region of the skeletal muscle cell. As it does that, it opens up voltage-gated calcium channels, allowing for the calcium that is stored within the cisternae to move into the sarcoplasmic reticulum and then wash through the cell itself, allowing for the interactions of the actin and the myosin. When we look at these cylindrical myofibrils, we have to remember that they can be made up of various filaments, what we refer to as proteins, in which we have three classifications. We have the contractile proteins. The contractile proteins include the myosin and the actin. We sometimes will refer to these as thick and thin filaments when we talk to individuals. However, we have to remember that we have to discuss them as either being myosin or actin within the contraction cycle. Myosin is about twice as large as the actin molecule itself, which is why it gets referenced as being the thick filament, and why actin is referenced as the thin filament. We have two regulatory proteins that come into play. We have troponin and tropomyosin. Troponin and tropomyosin is going to block the binding sites on actin, thereby limiting the interactions of actin and myosin. And then we have a couple of structural proteins that come into play, including titin and dystrophin, as well as the linking proteins. There are other structural proteins besides these, such as collagen and elastin, that will come into play when we look at the full muscle in terms of kinematics of large contractions. And so, if we put all of this together, we have to remember that we have this style of arrangements in which we have a central myosin polymer with its myosin heads, referenced as the cross bridges on the image here, surrounded by actin filaments in a 360-degree fashion. I'll show you what it looks like here in a second. Wrapping around the actin are the two regulatory proteins, the troponin and the tropomyosin. The actin itself is a string of globular proteins where there is a binding site on each of the globular proteins. Each binding site will correspond to a position for the myosin head to attach to. At rest, tropomyosin will be blocking those binding sites because of the conformation of troponin. The calcium that leaves the sarcoplasmic reticulum will be interacting with that troponin. When troponin is interacting with calcium, it allows for the tropomyosin to slide away from the actin molecule, thereby opening up those binding sites. So let's take a look at what this all plays out to when we look at the muscle itself. So the muscle itself is going to be comprised of various fascicles of muscle fibers. Within those muscle fibers, we have the individual cells. So we have the muscle itself is built of multiple fascicles. Multiple fascicles are built of multiple fibers. Within each of the fibers, we have the cells. In the cell, we have the functional unit, which is referred to as the sarcomere. The sarcomere is going to run from what's referred to as a Z-line or Z-disc to a Z-line or Z-disc. Within that, we have the interactions of the actin and the myosin proteins. It's done in such a way that the actin molecules will surround the myosin molecules in a cogwheel fashion. The actin will attach to the Z proteins, to the Z lines, via titin. That titin will link onto the collagen and elastin within the myosyns of the muscle, which are held in place by the dystrophin, giving the belly or the gaster of the muscle its shape. The arrangement of the actin around the myosin function in such a way that the globular heads of the myosin are able to pull on all surrounding actin molecules. If we take this 360-degree motion and lay it out as a flat image, we get something that looks like this, in which we have distinct regions within the sarcomere. Remember, we're going to go from Z line to Z line, or Z disc to Z disc for the sarcomere. Surrounding each Z line is an area known as the I band. The I band is the area where at rest, actin is the only protein that's present in terms of the contractile proteins. At rest, there's a region within the A band, the area of interaction, where we do not see any actin, and that's what's referred to as the H zone. During a contraction, the I band and the H zone disappear. They disappear as the myosin proteins will pull the actin proteins towards the M line. The M line is a set of myosin linking proteins that hold the myosin in place so that when they are doing their active pulling, the only thing that's going to move within the sarcomere is the actin. As the actin moves towards itself, the Z lines compress. As the Z lines compress, it pulls on the other structural connective tissue, causing the entirety of the fascicle to compress. As the fascicle starts to compress, it pulls on its connective tissue, causing the entirety of the muscle belly itself to compress into that contraction of the muscle.